Good afternoon. My name is Arun Bhunia in the Department of Food Science, food microbiologist. Our invention is how to improve drug delivery through epithelial barrier in the gut. And so we all recognize that oral delivery is the best way to deliver drug or take drug. We all take drug through oral route because it's safe, easy to swallow, minimal discomfort, and we all prefer that. Even a lot of injectable drugs are nowadays being converted for oral delivery. But there is a problem. Only a small fraction of drugs are being bioavailable when you consume through oral route. And among many factors, epithelial barrier actually plays an important role because a lot of drugs are not bioavailable because of the complex nature of the epithelial mucosal barrier. As a result, we lose a lot of drugs. So it costs really pharmaceutical industry quite a bit of money. And the market for drug delivery is really growing. And it's projected to be very high, and its overall growth is about 7.2% market growth is expected until 2021. So with that in mind, so how can you improve drug delivery? And our laboratory actually focuses on understanding pathogenesis of foodborne microorganisms which are taken through food. And we recognize that one of the protein produced by Listeria that actually increases epithelial barrier permeability. So this protein, when you apply in an in vitro cell culture model, once the protein is added, and if you add any drug analog, like in this case we are using FD4, and that permeability actually increased over time in the presence of lab protein. And also the delivery of this drug also increased with increased amount of lab being added to the complex. So we also proved that in our animal model, in a mouse, when there is lab is present and the drug delivery, in this case, we measured FD4 in the serum as well as in the urine, and you can clearly see the level of drug FD4 increased in presence of lab protein. And now we understand also the mechanism, how do they open the tight junction barrier. And lab interacts with heat shock protein 60 on the epithelial cell in the gut, and it activates NF-kappa B pathway. Also, it activates myosin light chain kinase, which really controls the integrity of the epithelial cells. So the epithelial proteins, which really hold together, these are moved away. As a result, the barrier opens and it allows the drug to pass through. And we also understood this tight junction opening is also very transient. It's for a brief period of time, once the drug is passed, and then it closes. So our legal landscape, we have already filed a provisional patent. And other factors I want to remind you is that this protein is non-toxic, it is safe, and as I said, the effect is transient, and it can be used for oral dosing of not only the pharmaceuticals or drugs, also the nutraceuticals or any vaccine candidate through epithelial barrier. So with that, we feel this lab protein or the peptide fragments has a great potential to improve epithelial gut barrier permeability, therefore help improve drug delivery through oral route. Thank you.